Hello and Assalamualaikum. Welcome to BIO4YOU. Okay, in this video, we're going to discuss about the deviation from Mendelian inheritance until sex-linked inheritance. This will be the last video for this chapter of genetic inheritance. Okay, let's have a look. Okay, the fourth one that we're going to discuss is polygenes or polygenic inheritance. Okay, so a character is controlled by the cumulative effect of more than one gene. Okay, usually to control one trait or one characteristics, we only require one gene. For example, gene for the color of the seed or the color of the flower, you only require one gene. But this one, many genes can determine one trait. So hence come the name polygene. Poly means many or banyak. So in this case, we have three genes that will determine one single characteristics. It occurs when one characteristic is controlled by two or more genes at different loci on different chromosomes. Okay, the main example in our syllabus is the skin color. Okay, it will show a bell-shaped curve called a normal distribution. Okay, and this will have the effect from environmental factors and could affect the phenotypes of polygenic inheritance. So when we try to plot the distribution of the skin color, it will gives you a normal distribution curve. Okay, where some is having a very fair skin tone, some having an intermediate brownish skin tone, and some having a much darker skin tone. So, skin color in humans is determined by three pair of genes. Okay, the character controlled by pairs of allele A, B, and also C, and at three unlinked loci. Okay, means at three separate chromosomes. The capital letters A, B, and C represent incompletely dominant alleles producing the dark skin. Okay, capital A, capital B, and capital C. Okay, this means that capital A, capital E, A, capital B, capital B, capital C person would be a very dark skin tone, while a small a, small b, small b, small c, okay, individual would have a very fair skin tone. Okay, and capital A, capital B, small b, capital C, small c person would have skin of an intermediate shade. Okay, so this linked gene, the genes are located on the same chromosomes. Okay, this one is slightly different than the polygenes. Okay, how does it differ is that they are always inherited together. Okay, and it acts as a single unit during meiosis, and they do not undergo independent assortment, i.e. it does not really follow the Mendel's second law, whereby in Mendel's second law we believe that each trait or each gene should assort independently, but not in this case. Why? Because if we observe here, gene F and gene G, they are on the same chromosome. Same goes with gene H and gene I. So F, G, H, and I, they will always be inherited together. Wherever F go, this G will also go because they are on the same chromosome. So this involves two characteristics determined by two genes. For example, in fruit fly Drosophila melanogaster, okay, we're going to observe the body color. The allelic pair is capital G and small g, where the capital G is for the dominant allele for grey body, and small letter G, allele small letter G, is a recessive allele for black body. And the wing size, the allelic pair is capital N, small n, where the capital N is for the normal wing, and for the small letter n is a recessive allele for vestigial wing. Okay, vestigial means slightly shorter wing. But these two pairs of allele located on the same homologous chromosome. So wherever G goes, N will also go. Okay. They will be inherited together. So this the representation of the diagram showing you the type of the wing shape and also wing size and also the color of the body. Okay. So this slide is showing you the comparison between the standard dihybrid cross and the linked genes, because they might tend to be confused together. Okay, so in the dihybrid cross, the two pair of alleles are located on different homologous chromosomes. You see, G and N is totally entirely on different chromosomes. Remember that each organism has some of organism have a lot number of chromosomes, some has only small number, but yeah, we have many number of chromosomes. And if they are gene G and N, they are on separate homologous chromosome, they can undergo independent assortment, i.e. they can arrange independently. But however, in linked gene, since they are located on the same homologous chromosome, so they do not undergo independent assortment. And the way the representation of the genotype writing is also different, where we're going to write capital G, capital N slash G and N. Why we need to do write in this way compared to the typical dihybrid, where we're going to put together G with G and with N, is because that this capital G and capital N, they will go and move together. Okay, so and it will be slash with the G and N for the other side. Okay, and it also gives you a different set of possible gametes. Okay, so this diagram is showing you, okay, on one side where we have the typical of dihybrid, the normal dihybrid cross, and this one is the linked genes. Okay, so in normal setting or in dihybrid cross, the gene are located on different chromosomes, so there is no problem in complying with the in the law of independent assortment. So you can have this type of gamete, GN, GN, GN over here. But however, for the linked gene, so we can see here after metaphase 1 and after meiosis 2, one cell, okay, or one gamete will produce only one set from G, capital G and capital N, and small letter G, small letter N, because remember, wherever this capital G goes, the capital N will also join together because they are on the same chromosome. Wherever this chromosome go, it will brings you both of these capital G and capital N. Okay. However, this linked gene, if there is a process of crossing over occur between these homologous chromosome at the between the non-sister chromatids, okay, linked genes can be separated. However, during crossing over between non-sister chromatid of a pair of homologous chromosome during meiosis. Okay, so now let us look at this example. So originally we have the capital G, capital N, small g, small n, they are on separate chromosome. However, capital G and capital N are to be remembered on the same chromosome. After the process of replication, you have the sister chromatids and sister chromatids over here. And during prophase 1, there is a crossing over occur, an exchange of DNA between these two chromosomes. You will end up with capital G, capital N, capital G, small n, and also small g with capital N, small g with small n. So you end up with different combination of chromosome. Okay, so this is in the case if without no crossing over, you will maintain as G and N. This one is G and N. I mean capital G, capital N, small g, small n for no crossing over. But for with crossing over, it produces new recombinant chromosome, i.e. new combination of chromosome. Okay, so this side. So this slide is showing you two scenario where for this linked gene with no crossing over and then with crossing over. Okay, so for no crossing over event, if no crossing over event takes place, you will end up with this type of gamete, GN, capital G, capital N, small g, small n. But if there is a crossing over, you will end up with this new combination. Okay, linked gene with crossing over. When we do a test cross, okay, between if there is no crossing over occurs, the F2 genotype ratio is 1:1. And remember, when we do a test cross, means we are crossing it with a homozygous recessive. And then when the test cross is done on F1 offspring, and if the process of crossing over occurs, so there is no definite phenotype ratio for the F2 generation, i.e. it will has 4 phenotypes, but the ratio is not definite, means we cannot plan out actual nice ratio for them. Okay, but generally in F2 generation there will be two large groups with a nearly equal numbers known as the parental phenotypes, i.e. having the same phenotype as the parent, maknanya ada ciri-ciri yang sama macam parent dia. And the other two small groups with nearly equal numbers, nombor yang hampir sama, known as the recombinant phenotypes, iaitu ciri-ciri gabungan daripada parents mereka, tapi tak sama macam parents mereka. Okay, so look at this diagram. Okay, so we are doing a test cross with this homozygous recessive, okay, with this parental genotype over here. So after if there's a process of crossing over occur, you have this set of combination of the gamete. These are all the sperm and this is the ovum. So we can see from this combination we have a G capital N, capital G capital N, and let's say it is fertilized with small g small n, it will gives you capital G capital N and small g small n. But remember, the way of you writing it is not capital G and then capital N with N like the dihybrid. No, you need to combine together G and N for capital G capital N, then slash with small g small n. Okay, and then you continue the process, and what you will notice later is that these two groups, these are the parental phenotype. What I mean by parental phenotype is capital G, capital N, small g, small n is exactly the same as this parent. Capital G, capital N, same with goes over here, and small g, small n. So this offspring is identical with this parent. However, this offspring, small g, small n, is identical with this parent. That's why it is called as parental phenotype. Same goes with this one. However, these two are called as recombinant phenotype because they are not exactly as their parents, neither similar with any of the parents. They are only a combination of the both parents. Okay, next we're going to look at the differences between Mendel's ratio and crosses that deviates from the Mendel's ratio. Okay, so if you do a test cross F1 with homozygous recessive, the dihybrid cross in Mendel's ratio will gives you 1, 1, 1, 1. And however, for linked gene with crossing over, the four phenotypes, the empat jenis itu has no specific ratio. Okay, and this one will obey, of course, the Mendelian law. This one is deviating from the Mendel's law for the linked gene. Okay, so this is what I meant earlier when two of the phenotype which are called as parental phenotype having two big numbers of about similar number 200 something, and the recombinant phenotype having much far smaller number and also about the same or similar number 40 something. So the F2 offspring obtained are as follow: grey body, normal wings, this number; black body, vestigial wing, this number. Grey body, normal wings is exactly as one of the parent. Black body, vestigial wing is also exactly like the other parents. So dua-dua ni adalah ciri yang sama-sama macam parents dia orang. Manakala grey body and vestigial wing, tidak ada parents dia pun yang ada grey body dan vestigial wings. And tak ada parents juga yang ada black body with normal wings. So these two, this one and this one is the recombinant phenotype. So for the recombinant phenotype, it is having much more for the smaller number. Okay, so in summary, for these linked genes, there are when there are four phenotypes in the F2 generation, two large groups and two small groups. So we can conclude, we know that this is a linked gene with crossing over. Okay. Next, we move on onto the sex chromosome. Okay, the deviations from Mendelian inheritance. Okay, in most species, sex is determined by sex chromosomes, i.e., sex means is gender, jantina. In human, two types of sex chromosomes exist: X chromosome and Y chromosome. X chromosome is having larger in size, whereas Y chromosome is much smaller than size. So of course, when you come in small package, smaller size, you lack some of the gene that is present on the X chromosome. So humans have a pair of sex chromosomes and 22 pairs of autosomes. So altogether 23 pairs. Okay, female has two X chromosomes, whereas male only has one chromosome and one X chromosome and one Y chromosome, gives you XY. So X chromosome contain many other vital genes for life. So for this sex-linked gene, the genes that we're going to discuss or we're going to study, they are located in these sex chromosomes, okay, i.e. on this X chromosome. Female may be heterozygous and homozygous individuals. And female, because female has two sex-linked allele, because female is having both X, okay? And male only has one sex-linked allele, so it is called as hemizygous. That is why males suffer more from the effects of sex-linked diseases and more often than females. The example of diseases is such as the hemophilia and color blindness in human. Okay, this slide is showing you X chromosome and Y chromosome compared to each other side by side. So definitely in terms of size, Y is much smaller. And on this X chromosome, the gene for hemophilia and color blindness is not present on the Y chromosome. Maksudnya tidak ada dekat Y chromosome. So by just having by just having a recessive hemophilia, okay, will cause the male to suffer the disease because there is no counterpart, tak ada nak lawan dengan dia dekat Y chromosome because there is no gene for hemophilia in Y chromosome. But in female, she can be a carrier. Carrier means they're being heterozygous. So she can have on one X chromosome having hemophilia, on the other X chromosome not having hemophilia, and she is normal. She's not a hemophiliac patient. She's only a carrier. Same goes with color blindness. Okay. So this hemophilia is the disease when blood does not clot properly, lack of the protein clotting factor, factor 8 or factor 9. Okay, these are types of protein found in our blood. Okay, the symptoms are bleeding and more susceptible to internal bleeding because the bleeding cannot stop. Okay, so for hemophilia patient is determined by two allele. This is how you write the genotype for hemophilia. Capital letter X with capital letter H. Okay, on the superscript. Superscript means position smaller size and on the top right corner. And X small letter H for hemophilia. Okay, so X capital H is normal, X small h is a hemophilia allele. Okay, so if the genotype, okay, so these are all in female, X. This one is in male because XY. So female can be a normal female if she is having both normal alleles, and she is only being a carrier if she's having heterozygous, and she only suffer from the hemophilia if she is a homozygous recessive. However, in male, the male is only a normal if the X chromosome is having a normal hemophilia allele. However, if it is having a hemophilia allele, okay, X with small letter H here, he will suffer from the disease, because even a recessive here will be expressed because there is no counterpart on the Y chromosome. Okay, so here are the list of inheritance that deviates from Mendel's law. The first one, codominant allele, represented by the example of human MN blood group. Okay, incomplete dominance shown by the antherinum sp plants, i.e. the snapdragon plants. Okay, and multiple alleles is shown by the ABO blood group in human, and polygenes such as in human skin color and also human height. Okay, linked genes, the body color and wing size of Drosophila melanogaster, and sex-linked gene, yeah, as mentioned as hemophilia and eye color blindness. Alright, that will be all for this chapter. Alright, again, please subscribe, drop a like, and share with your friends. Okay, and you can use this video always as tools for your revision, because the slides are really directly compatible with the slides, lecture slides that you have. Okay, so until next chapter. Alright, for now, thank you and bye bye.